The coursework is Gideon in class. Gideon is not in class. Is Daniel in class? Is Daniel in class?
Yes, ma. Good morning, ma. I just came in, ma.
Okay. So I want you to help me take care of the class. Where
Okay,
are
ma.
you now? Please don't. Okay. Unmute first before. Okay. Help me control the class. Thank you very much. Good morning,
Okay.
everyone. So we're welcome to, please, immediately, can you take the attendance now? Take a snapshot of the attendance, people online. Then I will tell you when next to take again. We'll be taking attendance twice. I think the next class, just because I want to know, are most of you available on campus so that we can have physical class? I want to know your thoughts on the general platform. I want us to optimize our time for class. So I think last week we touched about flavonoids, talk about what flavonoids are. We also talk about the sources of these flavonoids. We say virtually we get them from our fruits and veggies. That's why you always encourage to eat fruits and veggies. And we said that these flavonoids are antioxidants, meaning they fight oxidation in the human system. Okay, naturally, when a child is born, the child immune system is strong because they are natural antioxidants. But when we start working, start stressing, we have a oxidative reaction occurring, which is going to limit the antioxidant that is naturally in our system. That's why we need to add more by eating well to boost our immune system. So these antioxidants, they either enhance or mimic antioxidant enzymes. Remember, I said they are natural ones. So either they enhance the activity or they mimic it. Okay, they can either do by direct scavenging of the radical species. Now, remember the purpose of an antioxidant is to scavenge, to prevent or stop oxidation. So these oxidants can be metals. That's what I'll talk about metal chelating activity of antioxidants. It can also be by scavenging free radical species, radical oxygen species or free radicals. They can also do by repairing damaged cells. Cells that these antioxidants have really damaged. Okay, that's what I'll talk about cellular repair. Remember, life is on cells. You are as healthy as your cell. From cell, we'll talk about system. From system, we'll talk about organs, cell, organ, system, and whatever, and process. So from your cell, you go to organs. So when your cells are bad, it affects the organs. When your organs are damaged, it affects the system. So when the systems are damaged, that's when you see somebody, they say, this one is, the person has died. Like you're talking about when your cells are bad, from there it starts affecting the kidney. Kidney is the organ. How can you be behaving like that, Bukola Adebisi? Anybody that disturb my class, I'll just note down your name. I will not disturb you. On the day of reckoning with you. Don't disturb my class, I beg. I like when I'm flowing in this natural product. I don't like distraction, please. Okay, we're talking about the antioxidant activities. Okay. And at times, there's a way you take iron, like blood tonic, it becomes excess, it becomes poisonous. So at times, some of these antioxidants can take away some of the metals that are poisonous in your system, we call them, they chelate it. They bind all these metals and mop them off your system. That's why I see at times all these herbs. When you take herbs, they said roughages. So they purge you. When they purge you, they are taking out some of those things that are not supposed to be in your system. So by purging, at times purging is detoxification is good. But when it becomes excess, when you're eating and your food are not digesting, you're just purging, that's when it becomes sickness. So it's two ways. So talking about flavonoids. Another thing we're supposed to know about flavonoids are I talk about that we can obtain them from higher vascular plants, particularly in the flowers, in the leaves, and the bark. We also see them in fruits, grains, and nuts. Remember, I said when you talk about fruits, these flavonoids are many on the skin. And that's why I said if you're eating something like granite, or especially blood type A, is that granite is beneficial for. Eating granite with the skin, with the peel, is more healthy than peeling off your granite skin. When you're taking your something like apple with the skin is healthier. The problem we're having is all these chemicals. That's why people peel. When you peel, you're taking most of the nutrients. Just like talking about onion, the peel of onion that we discard is actually beneficial. So some of the peel we discard in the dustbin, we are discarding the most potent aspect of that fruit or veggies, which is the flavonoid. When you talk about red wine, what make red wine very healthy for the heart is because of the berry, which was fermented, making it to contain loads of flavonoids, which is antioxidants. So you also talk about, when you're talking about your vitamin C's and all these things, they're all antioxidants, right? If you go to something like all this food that has vitamin C, you can see they are colored, they are very colored. Even our carotenoids, which we see in carrots, are also colored. So some of these colors, fruits and veggies, are potent source of your flavonoid or your carotenoid. At times, vitamin C, as you can see in your orange. So in today's class, so in this particular section of this class, we'll be looking at the introduction, what are flavonoids? We are looking at their structures, their occurrence, their nomenclature. Now, we are going to look at the flavonoids in general. We are going to see catechins, which is flavanols. We are going to see condensed tannins, which is called proanthocyanidins. So we're going to also see the functions of these flavonoids in vitro. This is in vitro, is in the cell. Then we're going to talk about their radical scavenging potential, their redox properties, and the mechanism of their radical interactions. So that's what we'll be looking at in the course of this flavonoid slides. We've talked about the flavonoids are secondary metabolites. We'll talk about that, and we'll talk about the most of them, the higher the number of hydroxy, the more the antioxidant potentials. So when you talk about flavonoid, you're talking about polyhydroxyl compound or polyhydroxy aromatic compounds. Okay. Then you also talk about that these flavonoids, they have function both in plants, okay, and also in humans. Now, example of whenever you're talking about somebody talking about green tea, what they're talking about green tea is because of the antioxidant potentials of the green tea. Whenever you're talking about grape seed or their skin or almighty cocoa powder. I remember those days when cocoa powder was raining. If you've not taken cocoa powder, you know, in Nigeria, anything that rains and everybody forgets it. In the era of something like moringa, everybody, if you've not eaten moringa, you're not there yet. But now everything is like people see moringa and take their eyes off. And this is very bad because some of these things that we are raining then are actually very beneficial. But when you neglect them, does not mean they've stopped being beneficial. But there are some people that are also optimizing the benefits. So whenever you know those things, there's a saying in the Bible that say, happy are you if you do them. So you must make sure you're a doer, not just that you know. We all know that cocoa powder is healthy. How many of us take cocoa powder? we prefer taking Milo than cocoa powder, even knowing that Milo we are taking is bad. But because of our sweet mouth, we don't want to stress the body. But at the long run, what we are eating is what will also kill us. So that's why some of these things that you know about, optimize them for your health. Okay. Now, this flavonoid, they help us to screen this, they shield us from UV light. Okay. They have scavenging, they have astringency properties. That's one of them. Like some things you take. What will I remember now? That you start feeling this mental feel. That's astringency properties. We can also talk about the pro-anthocyanidin in green tea, which is Camellia sinensis. This is what makes green tea good for diabetes because of this pro-anthocyanidin. Okay. And others. Something like cocoa powder is also very good for diabetes. So if you have history of diabetes or have anyone that have them, please recommend cocoa powder and green tea for them. It's very, very helpful. And if you have the history in your either your father, your mother, or your relative that is know that it runs in your family, please be careful the way you take all those beverages. Instead, go for natural. Okay. Now, antioxidant potential of flavonoids can reside both in their radical scavenging activity or meta scavenging. So it means that flavonoids can work and can exhibit the antioxidant capacity either by scavenging or by chelating. But the one that is much more prominent, and we're going to discuss more in this course, is the free radical scavenging potential of flavonoid. Okay, we are talking about flavonoid being polyphenolic compounds. When something is polyphenolic, means that it has more than one phenol in a compound. Okay, then flavonoids comprise of 15C carbon, okay, and 2 rings. And these are connected by a 3C link, which is C6, C3, and C6 bridge. Now, if you look at the ring we have A and B, right? And we have 2 aromatic rings. And this ring is connected by 2 C's. You can see that C. Okay, C6, C3. You can see the 6 connected to 6 and another 6. So the 2 rings are C6 and they're linked up together by another C3. Okay, so that's why we have the ring, and this ring is called flavan. And this flavan ring is what like a moiety for every flavonoid you are going to have. So every flavonoid you are going to be talking about is going to have this ring A and ring B. So what will differ is the ring C, which is one where the 3C is going to form. So that's why we call it the basic moiety or structure, basic skeleton of flavonoid. This flavonoid comprises all compounds whose structure are based on flavone. Look at the structure of flavone. You can see flavone contains ring A and ring B. The difference between ring A and ring B is that my ring A is connected to ring C, while ring B is like an attachment to ring A and B. So the essence of this is for you to understand how these structures are. So whenever we are in the class, just make sure you are conversant with the structures. So whenever I want to talk about flavonoid, the basic moiety I will look at is 2 rings, okay, connected by the 3rd ring. Ring A and ring C are connected together. So it means that they're sharing 2 carbons together. While ring B is different, it's like a substituent. So the basic moiety for all flavonoids is called a flavone. We look at the structure, core structure, and nomenclature of these flavonoids. Look at charcoal. You can see my A and B are untouched. What happens changes is my C. On my flavone, flavone is a ketone. That's why it's called one. And if you look at all the structures, you can see that eta ring is permanent in all. So what changes is the carbon That's the ketone functional group. That the other one that bears the ketone, right? So if you go to the, we are talking about the carbon number 1 of C ring. So this carbon number 1 of C ring, carbon number 1 of C ring is what actually changes, okay? Because my eta ring is constant in all the structures. So I want us to go together so that we can go slowly. So look at my flavone, look at my dihydroflavanol, look at my flavan-3-ol. My eta is still intact. My A and B are still intact. Look at my flavone. Flavone and flavanone are not the same. Look at flavan-3-ol. That ol means that there is hydroxy group. Look at anthocyanidine. Look at isoflavone and neoflavone. So these are the core structures and nomenclatures of most flavonoids. A converses to a heterocyclic ring C and an aromatic ring B, and most often attached at C2 position. Okay, chalcones is only one that has an open ring. Others are closed except chalcone, right? Now, this is flavonoid structure. Now, previous slide talking about our core structure, core. That's the basic structure. So whenever I'm talking about any structure that has to do with flavone, this is a basic structure. So if I'm talking about flavan-3-ol, this is a basic structure. So see, that's a basic core structure. Does not make it a specific name of an class of this flavonoid. So if I have flavone, my flavone might have another name, but this is a basic structure of flavone. So let's continue as we look at the other slide. Got to see now. Now we have flavanol. In flavanol, we have hydroxy group on ring A, 2 hydroxy group on ring A, and we have R1, R2, R3 on ring B. Only 1 hydroxy group on ring C. That's of flavanol. Now look at flavonol. Flavonol. If you look there, you can see difference between flavanol and flavonol. Difference between them is in flavonol, I have double bond on ring C and I have a ketone functional group. That's just different between them and my flavanol. So if I know flavanol, I can know flavonol because I'll just add double bond, okay, and I add my ketone functional group to form my flavonol. Now look at flavonone. Difference between flavanol, and flavonol is that the double bond is removed and hydroxy group is removed. So the same skeleton of flavanol, flavonol, and flavanol, they are similar. Difference is either you're adding or you're removing. So with one, I can be able to draw three. Three are structures of flavonoid. So I can draw flavanol, I can draw flavonol, I can draw flavanone. 3. Okay, the only one that is different, then look at anthocyanin. What's different between my flavonol and anthocyanin? If you look at the difference, you can realize that my difference is that as I'm removing my ketone functional group on flavonol, I'm going to add conjugated double bond. So I'll add conjugated double bond, then remove my ketone functional group to move from flavonol to anthocyanin. Then in my anthocyanin again, I have additional hydroxy group in my ring B. But remember, my flavonol, and flavonone, they don't have any, they have just R1, R2, R3 on the ring B. Then look at the isoflavone. What's the difference between my isoflavone? The isoflavone is another structure entirely. If you go to that core structure, where we talk about isoflavone, look at the structure of isoflavone. Do you have isoflavone here? Yes. So you can look at the core ring of isoflavone, and also look at the typical example of isoflavone given. So these are flavonoid structures. So the more we go, the more we understand the examples of these flavonoids. These flavonoids are classified by virtue of their linkage of aromatic ring. That is benzopyrone. This benzopyrone is ring or is the second as ring C. This group of natural product may be divided into three classes: true flavonoid, isoflavonoids, and neoflavonoids. Classification of flavonoids are into true flavonoid, isoflavonoid, or neoflavonoid. Now, look at them very well. If you look at the true flavonoid, you can see that true flavonoid, the moiety, it has this benzopyrone, right? And it's on carbon number 2. We call it 2-phenyl because the phenyl we are talking about is ring B. Remember that ring B is like a substituent, so we call it a phenyl. So and this is attached on the benzopyrone. That chromano moiety is attached to that chromano moiety on ring 2. So that we'll call it 2-phenyl benzopyrene. If you look at this one, it's attached on 3. We call it 3-benzopyrene. The other one on 4, we call it 4-benzopyrene. So for true flavonoid, we call it 2-phenyl. Then for iso, we call it 3-phenyl. For neo, we call it 4-phenyl benzopyrene. So 2-phenyl, 3-phenyl, and 4-phenyl, indicating true, iso, and neo. In that slide, we're supposed to add phenyl, okay, in the benzopyrene. Okay, let's look at this classification of the true flavonoid. Now, the true flavonoid are one. They can either be chromane derivative or chromone derivatives. Chromane or chromone. Remember, they are meant to be in 2-phenyl benzopyrene. So they can either be chromane or chromone derivatives. If you're talking about chromane, we're talking about the catechin, which is flavon-3-ol, or leucoanthocyanidin, or anthocyanidin. You're going to understand these things better as we proceed. But when we're talking about chromone, we're talking about flavone. Now, from this, you can understand the chromones. That was something you're going to know in their structure, as you're going to see. How I can distinguish between my chromane and my chromone. My chromones are the flavonones, flavonols, flavones, flavonols. The other one is flavanol. This one is chalcones, dihydrochalcones, and arones. Okay. So the first one is flavanol. It's NOL. Flavonols. Not N-O, not NON. NOL. Flavanol. Nols. It is L. So you correct it. This one is different between flavanol and flavonols. You can see difference is the O after V. Va and vo. So we have flavanols, we have flavonols, we have flavones, we have flavonols, we have chalcones, we have dihydrochalcones, and we have arones. So these are the chromone derivatives. Under chromane, we just have catechin, leucoanthocyanidin, and anthocyanidins. What's the sense of all this? Now, let's look at it now. Under flavones, the example of flavones. Now, there's something we need to understand. Whenever you see a sugar moiety, you know we are dealing with anthocyanin. When there is no sugar moiety, it becomes cyanidin. Okay? So we have sugar moiety and non-sugar moiety. So anyway, whenever you see sugar moiety, talk about sugar. So look at Neragenine when my arrow is H, when there is no sugar, but it become Neragenine when I have sugar moiety. So my sugar moiety, that's why the essence of this sugar moiety is why some fruits are very sweet. So not all flavonoids are sweet. Some flavonoids can also not be sweet. Some are meant to be sweet. The one that are sweet is because they contain sugar moiety. You can see this one have rhamnoglycosyl. Okay, this one have galactosyl. This one have glucosyl. So these are what makes this flavonoid to be sweet. Look at quercetin. Quercetin is what makes taking apple a day keeps the doctor away. It's because of the presence of quercetin. So if you want to draw the structure of quercetin, whenever you see your R, you're going to replace it with H, meaning that we are going to have alcohol instead of ether on that, instead of methoxy on carbon number 2 of the chromona or chromo, that's ring C. Okay, that's then that. So because this issue of quercetin is very, very important for you to understand quercetin. Okay, and quercetin is a group of flavonols. So if you're talking about example of flavonols, you talk about quercetin. If you're talking about example of flavonols, you're talking about hesperidin or hesperidine. What differ between them is non-sugar moiety or sugar moiety. That is. Then if you're talking about flavonols, now so we have two flavonols. The upper two are two flavonols. So because if you look at them, they don't have any hydroxy group on that C ring. Okay, so that's why we're calling them what? Flavonols. The others have hydroxy group, like quercetin has hydroxy group on that C ring. We call it flavonols. Okay, now whenever you want to understand this structure, what you ask yourself, what is missing, what is added, what is removed between naringenin and hesperidin. The difference we can see is on the B ring. On that B ring of naringenin, we have hydroxy group alone, while in the one of hesperidin, we have methoxy and hydroxy. Every other thing is similar. So that's how you study these structures. When you look at the structure, look at what is missing. You might be asked to give example of 3 flavonoids or 2 flavonoids with structures. So any flavonoid you want to know, understand that flavonoid and being able to draw the structure very well. You can be asked, okay, class of flavonoid with the example. If I want to give example of flavonoids, I can either use my naringenin, or I'll use my hesperidin, or I'll use my naringenin, anyone. But I should understand who the class they belong. So you should be able to know flavonoid by their class, an example. Okay, if my class is flavonol, either I use rutin, or I use isoquercetin, or I use hyperoside or quercetin. Any one I know, just be sure that you know one and know that one very well. No need of you learning everything, because you cannot, it's not possible for you to know everything. So anyone you're going to learn, be consistent. That's the sense of practice. Practice them. Okay, I want to study class of flavonoid, an example. I bring up my Bible, I write class. Okay, flavonol, example, structure. Flavonol, example, just like that. With that, by the time you do it 2 times, you'll be fine. Remember, under the core structure, we talked about anthocyanin. Let me go back to that core structure where we talk about anthocyanin. Now we'll talk about anthocyanin, and I will talk about that in that anthocyanin, you can see the anthocyanin has ring, the 3 rings, okay, but there is hydroxy group and there is conjugation in C ring. Let's go back. So talking about that, these anthocyanins, they are one of the groups of flavonoids. So among the class of flavonoids, we have what? Anthocyanins. We can then, whenever there is sugar, anthocyanin and anthocyanidin, they are also the same. Okay, one is have sugar, one does not have sugar. The one that have sugar, that is sugar free, we call them anthocyanin. While the one that have sugar, we call them anthocyanidin. Okay, they are natural pigments. Like I said, they can be glucoside or a glycon. Glucoside is one that have sugar, and the one that does not have sugar, we call them sugar-free or glycons. These anthocyanidins are water soluble. Flavonoids are water soluble. Remember they are polyhydroxy. So they are water soluble. They have colors. They are responsible for colors. All these red, violet, and blue colors. You know, some of the potato you see there, they have all these violet color, the blue colors, all those colors. When you see this red color, they are not just flavonoid, they are anthocyanins. When you see violet color that are very strange in some fruits, they are not just flavonoid, they are anthocyanin. So they are class of flavonoids, okay? And they are amphoteric in nature, meaning they can behave as an acid or they can behave as an alkali, depending on the media on which they are in. So this anthocyanin, their acidic salt is red, their metallic salt is blue, and their neutral salt solution is violet. That's why they have red, they have blue, and they have violet. So in acid, they are red, in metal, they are blue, and in neutral solution, they are violet. Meaning in acid, they are blue, in base, they are blue. In acid, they are red, in base, they are blue. In neutral solution, they are violet. Okay, these colors are due to metal chelation. Remember, I said that flavonoids can act as scavengers or chelators. They can chelate metal or they can scavenge free radicals. So by virtue of anthocyanin, anthocyanin can work as a chelating agent. So the color we are talking about is due to the metal chelation that is affected when molecules that contain two hydroxy groups in auto position. So because of this, that is why, remember, if you look at the structure, we talk about there is conjugation of one hydroxy group in that chromo. So because of this change, that is why there are always different color in different media. Now, importantly, we have derivatives of this anthocyanin. Most of the derivatives are 357, which has 2-phenylbenzopyrene chloride and sperrine compound. 357 is like a moiety for all the derivatives we are going to see. We are going to have about 3 derivatives, but this is a moiety. So look at it. Look at the basic moiety 357 trihydroxyflavonium chloride. Look at the flavonium chloride written, and look at the 357 trihydroxy. So these are the basic skeleton of all the 3 derivatives of anthocyanin we are going to talk about. Remember, anthocyanin is a type of flavonoid. So they have 3 derivatives. Remember, this derivative is by virtue of the metal chelation that occur. Okay, so this metal chelation is going to cause different color change, which brought about 3 derivatives. There are 2-phenylbenzopyrene chloride is the parent compound. That is also what is called flavonium chloride. Flavonium chloride is the same thing as 2-phenylbenzopyrene chloride, so they are the same thing. So whenever we're talking about, them as the moiety. The moiety, what is moiety means that In every of the derivative you are going to see, 357 must be present as a trihydroxy group. So what would differ is any other thing that will be added in each of the derivative, which you are going to see in the next slide. Now look at this. This hydroxylation pattern, remember we're talking about what happened when this color change occurs. So this hydroxylation pattern is going to fall into three groups. But look at the first group. You can see my 3, 5, and 7 is constant. What I added is just 4 prime. The prime, okay, let's go to the previous slide. If you look at this slide, you can see that the ring of B is a ring of prime. Now, carbon number 1 is a linkage where B is linked to C. So we're going to go clockwise to have 2 prime, 3 prime, 4 prime, 5 prime. So whatever that is affecting affects the prime. Let's see how it goes in the next slide. You can see the first hydroxylation pattern we're going to form in anthocyanin is called pelargonidine. In this pelargonidine, we have 357, which is the moiety, and then we have additional 4 prime. So difference between my moiety, basic skeleton of this hydroxylation pattern, and pelargonidine is 4 prime of hydroxy group. Remember, we're talking about this hydroxylation pattern is where hydroxy group. The more the hydroxy group, the darker the color or the brighter the color. Okay. Now look at now, because of this hydroxylation pattern, we are going to have orange, red, and scarlet flowers. So this is why we have red rose and the red diala. So this is by virtue of this pelargonidine hydroxylation pattern, which is 3, 4 prime, 5, and 7. So the question is, what are the hydroxylation pattern in anthocyanidine or anthocyanin? Cyanidine. Cyanidine, I'm having 3 prime and 4 prime. Remember, in my pelargonidine, I have only 4 prime. That's cyanidine. I'm adding another one, which is 3 prime, making it 3 and 4 prime. Or the skeleton A and C remain
Really one place I'm
constant.
seeing you. Really one place I'm seeing you.
So an example is, this is found in bluish red flowers, like blue cornflower, red roses, and red dahlia, and red dahlia. So these are the presence of cyanidine. If you look at this thing, remember I talk about nin and din. Whenever you're talking about nin, I'm talking about din. So cyanidine is a hydroxylation pattern that have penta, that's 5 hydroxy group. While my pelargonidine have 4 hydroxylation pattern, that's 4 hydroxy group. Remember, the basic moiety has 3 hydroxy group. That's 3 hydroxylation pattern. When we come to definidine is going to have 3 prime, 4 prime, and 5 prime. Okay, so making it 3 plus 3, making it hexahydroxyl flavonium chloride. The basic moiety is tetra. The basic moiety is tri, while pelargonidine is tetra. Penta is cyanidine, then definidine is hexa. So you should be able to understand this and know the difference. When you're talking about din, it means there is no sugar moiety. When you're talking about various sugars have been found in anthocyanin, The most common are glucose, galactose, and rhamnose. So whenever there is DIN, there is no sugar moiety. So whenever there is no DIN, there is sugar moiety. So whenever we are talking about a reaction that has to do with sugar moiety, that's why in subsequent slide you're going to understand that when there is hydrolysis, it occurs by cleavage on that sugar moiety. Okay, but whenever we're talking about DIN, it is no sugar moiety. So the anthocyanidin we are talking about in the hydroxylation pattern does not have sugar moiety. How do we extract this flavonoid? Especially for people that will be doing natural product in their research. Normally, ethanol or methanol are often used in extraction of flavonoid. Okay, then when you're done with extraction, then we can treat with chloroform. The essence of chloroform is to remove something like chlorophyll, carotenoid, and waxes. Then before you purify the water residue with diethyl ether, ethyl acetate, propanol, or butanol. Now, from experience, ethyl acetate contains so much flavonoid from the research I carried out. Okay, and some of these flavonoids can be validated using chromatography. When you do chromatography, you're going to see some colored bands. Some of these color bands can only be detected using UV light or iodine tank. So flavonoids can be obtained by nature by using alcoholic extracts. Then they start doing separation. We call it gradient separation. In gradient, I start from non-polar to remove all the lipids. Then I can use mid-polar like diethyl acetate. Both diethyl ether can also do the work of removing lipids, okay? Then before I add my ethyl acetate to take out some flavonoids, propanol and butanol are going to take out most of the flavonoids. So all the flavonoids that are polyhydroxy must be taken out by something like butanol. And some of them, if you're doing column or you're doing separation, they're always difficult to separate because they are polyhydroxy. So they're always forming a tail. If you have done column before or you've done TLC, realize that your compounds are tailing, the bands are not separating, they are tailing. It's because of so much water, polyhydroxy groups, presence of flavonoid in your extract. How do we identify this flavonoid? You can identify flavonoid using tests like cyanidin test, testing with boric acid and citric acid, azocoupling, reaction of alkali. At times in the lab, when you're doing your phytochemical screening, you can see the reaction with sodium hydroxide. You can see the reaction with iron 3 chloride because to dictate the presence of these flavonoids. So this is a table that talked about flavonoid and the test. This is what they call phytochemical screening. Phytochemical screening is what you observe when you're doing your reaction. So it has to do with color change. So the color change will tell you the class of flavonoid you're dealing with. But naturally, when you're dealing with flavonoid, that's something that's always possible. When you're reacting, at times with sodium hydroxide or potassium hydroxide, you always see this yellow color. At times it might be somehow as if it's turning green because your plant is already green. If you're using leaf part of the plant, you realize that your color become greenish yellow. Okay, depending on the nature of your extract. So the color will tend towards the extract. You can also use iron 3 chloride. You can see at times you're going to form something like reddish brown or brown. Okay, so lead acetate also can be. So these are the reagents we can use to test the presence of flavonoid in the laboratory. Any questions till now? I'm not going to rush this class because there are most ones that are very, very important. So I'm going to finish it on Wednesday, but I'm going to end the class in this analysis. How do we analyze presence of flavonoid? Now, when you're talking about gravimetry methods, gravimetry method is something you can use for crude flavonoid. You want to know whether there are flavonoid in a sample after your extraction, okay, I realized that okay, I've removed my lipids using diacyliter on normal hexene. I've used my ethyl acetate to obtain some flavonoid. I've used my N-butanol to obtain remaining flavonoid at the end. When I evaporate my solvent, the crude flavonoid I measure. I'm going to weigh. So different in weight is by gravimetry, by weight. It's a crude method. Titration is more specific than gravimetry. You can use okay, fluorimetry, polarography, phytocalorimetry, but the most important method of obtaining this flavonoid specifically is spectrometry. Spectrometry is going to tell us the nature of the flavonoid, the name, also tell us the structure of the flavonoid involved. Now, whenever you want to identify the presence of flavonoid, you can use UV. UV is going to help you to see the visible regions in terms of their color. In your UV, you have the lower wavelength and higher wavelength. So depending on the nature of your flavonoid, you're going to observe them using UV light. So that's the end of today's class. As we take our classwork, if there's no question. And if there is question, unmute yourself and ask your question. Question, we're not going to have so much question here. Don't know the last question I gave us. But question number 1 is that I don't know whether I've asked question. Daniel, have I asked question of defining antioxidants?
No, ma.
Okay, the questions number 1, what do you understand by the term antioxidants? Number 1, what do you understand by the term antioxidant? Number 2, what is the mechanism of flavonoid as antioxidants? What is the mechanism of flavonoid as antioxidants? What's the mechanism of flavonoid as antioxidant? That's number 2. Daniel, taking?
Yes, taking, ma.
Okay, number 3 question. Okay, number 2 question. This is number 1A and B. Number 2A. List. Okay. Name 2 classes of flavonoid with the examples. Structure and name. 2 classes of flavonoid with examples. Or let's make it 3 classes. 3 classes of flavonoid with examples. That's 2A. Then 2B. There are 2 derivatives of 2 flavonoids. There are 2 derivatives of 2 flavonoids. Mention them with an example each. Mention them with an example each. A natural pigment that are natural pigment flavonoid, or that is the flavonoid that is known as natural natural plant pigments are called dash. The natural plant pigments are called dash. That's number 3. Natural
Sorry,
plant
ma. We have
pigments.
2. Sorry, ma. We have
No,
2A and 2B. So the next one is 3, right?
3A. The
Okay,
natural
natural.
plant pigment is called dash. The natural plant pigments are called dash. The type of flavonoid responsible for red, violet, blue colors are dash. The type of flavonoid responsible for red, violet, blue color are dash. That's 3B, right?
Yes, ma'am.
Sorry, ma, can you please repeat the question again?
Which of the questions?
The previous question.
Daniel, can you help us?
The type of flavonoid responsible for red, violet, and blue color are dash.
For red,
Red, violet, blue color are dash.
Okay, number 3 question. Number 3 what? Number 3 what are we?
3B, now it's 3C.
Okay, let's do that 3A, I and II. So let's do 3B now. 3A, I and I, and then 3B is there are dash hydroxylation pattern in anthocyanin. Mention them with structures. Mention them with structures. So anyone you're going to mention, you tell us their general name, the IUPAC name, and their structures. The hydroxylation pattern in anthocyanin. Mention 5 tests you can use to identify flavonoids. Mention, that's C now. Mention 5 tests you can use to identify flavonoids. Under that CII, what are the methods of quantifying flavonoids? What are the methods of quantifying flavonoids? Any of the methods, at least you should be able to mention like 2 methods of you can use to quantify flavonoid. So, like the norm, by 11:59 today, you should be able to submit your classwork. I'm going to share the slide. I don't know whether I've shared this flavonoid slide. I want you to go through them, read before you answer the questions. Don't copy from your neighbor. Just make sure you have an interaction with your slide. If it's possible, if I were you, when I'm reading, I'll be making notes. Because virtually, I want to start reading for my exams now. I want to start reading for my test now. So go and get an exercise book, might be 40 leaves, minimum of 40 leaves. If you can afford 16, is better. So after each class, go and make note of that particular class. Then after making the note, you answer the classwork. That's why I gave you that period, that luxury of time. You will do the classwork yourself, then submit. If you can follow up this way, there's no how you don't have this particular course, nothing less than 80%. Because on the day of test or exam, you're revising. You're not going to re-slide again. You're going to go and bring that jotter and revise. And whenever you have in your private timetable, you have 435, what you're reading is that jotter, because from the slide, you made your notes already. So you're good to go. By the time you finish reading that your jotter 5 or 6 times before test or exam, even if your village people follow you, you must have a very good day. Okay? Hope there's no question. If there's any question, I'm available to answer your question. Because I have lab by 11, I should be ending the class now. Thank you very much. See you in next class. Please submit your classwork. Daniel, I forgot, supposed to have taken. Please, let's take this snapshot again for the attendance. The last one now. Take it now. So the last people that started now are 61. That ended with us at 61. So take it and share with me. Thank you very much. I wish you the best. Do your classwork very well. Study and make your notes. I'll see you on Wednesday.